Thank you, the organizers, for the invitation and Irene for the kind introduction. And I'm happy to see all of you here at 7 p.m. in a Friday. So thank you very much for your audience. Tonight I'm going to talk a little bit about artificial intelligence for image analysis and how the things can go wrong when we are doing this. And I will start my talk telling that the AI is not a quick fix for image processing or image analysis, and I will tell you why. So we spend hours to days just acquiring data and annotating data, and after that we spend hours to days to train these models. The predictions can take seconds to minutes depending on what you're doing, and analyze the results of your model also can take seconds or hours. So AI is not the easy button that you can just press and have your results, but once it's done, can save you a ton of time. A great example of that is this cell segmentation approach, where we compare the manual annotation time to a cell pose model inference. So for annotating this kind of data manually, we should take about 11 minutes and to segment this same data using cell pose, that's a kind of a deep learn model or software, we're gonna spend just a fraction of a second. So really works, and you should really use if you know how to use and if you have a good model. To make the things clear, artificial intelligence is a computer system able to perform tasks normally requiring human intelligence. In the machine learning field, it's inside the AI thing and gives the computer the ability to learn without being explicitly programmed. And the deep learning version of this AI thing uses these machine learning algorithms in a brain-like structure, logic, and they are called artificial neural networks. To make it simple, we have here an input that's a car. And for machine learning, what we do is extracting the features. So we extract all the information that we know about the car. So this car is blue, this car has 2 wheels, or this car has 2 windows. And then we give all this information for our computer that will, using this neurological network, will classify this image as a car or not a car. For the deep learning task is a little bit more complicated for the computer, not for us. So we have the same input, and we give all the tasks for the computer. So the computer will perform the feature extraction, and then we'll classify this as a car or not a car. And today I'm talking about bioimages, and this is no more than biological information given by numbers, as mentioned before. So the images are composed by the raw data. That's just the matrix of pixels that compose and tell us the story about this image, and the metadata. That's super important in cell biology or every field that we are gonna study with images. And the metadata is any kind of text that provide additional information for us about the image. If we have an image, we can process this image, and the image processing is any form of data processing for which the input is an image and the output will not necessarily be an image. And here we have 3 examples, where here in the first example we have an input as an image. So this is a phase contrast microscope image of some cells, and the output of this image processing task are the coordinates of each cell. Here in the middle, we have an input as an image again, and the output is also an image. So at the left, we have the raw data, and the right, we have the label image, so telling us each of the objects that we have in this image. And we also have here an input as an image again, because we are image processing things, and the output it's a label. And here we have 2 examples of these infected cells or not infected cells. So here we are talking about image processing, and when we talk about image processing, we are talking about tasks that we can do with images like drift correction, denoising, improving sample resolution, or even artificial coloring some organelles, for example. And I will tell you that the image processing tasks are the playground for deep learning. We can probably do whatever we want in image processing nowadays using deep learning tools. And to exemplify what we can do, so here for the supervised convolutional neural networks, what we need to train, or how this architecture works. We need a pair of inputs and outputs. The outputs here are the ground truth, so these input images are processed by our architecture or our neural network, and this neural network will give us an output. This output will be compared to the ground truth that we provide to the system, and this will result in a loss function. This loss function will be used to modify the weights in our convolutional layer. That's the simple way to tell you, and this is the hard way to explain. But here is just an image segmentation convolutional neural network architecture that's called the 2D UNet. And to make it simple, we have here an image, and this image will be convoluted or will be filtered. So this will generate new images here, and in the network, these new images will be downsampled and on again, depending on your network. And after that, this convoluted new images will be upsampled and convoluted again until they reach the same size of our input. And how do we know that the system works? We need to verify that the model is doing precisely what we want. In this case, we have phase contrast cells here, and we want a model that recognize or segment our cells. And clearly here, the results of the model 1, it's not doing what we want, but the result of the model 2 is doing exactly what we want. That's segmenting these phase contrast cells. Our eyes here can do pretty nice job. We can see here what's going on and which model is better than others, but there's a bunch of flavors of metrics that we can use to quantify these, and you should use that. So the first problem when we are training new models or new models is inconsistent annotations. So here in this case, we want to quantify the protrusions or the number of protrusions in this time lapse of these cells. And if I ask you, all of you, to annotate where is the protrusion and where is the cell body here in this cell, I will have sure that any of you will point out where is the protrusion and where is the cell body in a different way. But they had this data and annotated like that, and they used this to train a model, and the result was this one. Okay, the model were able to recognize the cells and some protrusions, but the model didn't give me or didn't give them the number of protrusions for each cell. So what they had to do were to create a new imaging processing approach where they segment the entire cell and they point out just the tip of the protrusion. So training this new model, they were able to recognize and to count the number of protrusions in different cell shapes, like we see here. Another problem during training of models, it's image quality. And that can be really tricky because biomedical images can get really large, as Greg mentioned earlier, up to terabytes or petabytes. And during training, we have a major limitation, that's the GPU memory size. So we need to fill the the image inside our GPU, and that's our major limitation. So here they wanna segment cells in mitosis, and they have this huge image, about 7,000 pixels by 7,000 pixels, and normally this kind of huge image doesn't fill in GPU memory well. So one strategy is just to crop this image in 512 by 512 with the correspondence ground truth of this image. So they did that. So this new small image could fill in the GPU, and they trained the model, and this was the result of the model. Empty masks. They were not able to recognize any of these mitotic cells. Why that happens? Just because they forgot to see the receptive field of the network, so what the network sees. As I mentioned before in the beginning of this talk, we have a bunch of downsampling going on in the network, and the receptive field for this particular network was just this region of the cell. So the network was just seeing this, and this was not enough information for the model to recognize this model. So when I started with this bioimage analysis things and starting to apply AI and deep learning to my image analysis problems, I thought, okay, AI can solve everything for me and will be super easy. And yes, they can solve, but I forgot to think that I need to train a machine to perform this task. So here in this case, just to simplify for you, we have this kitten cat, and I want to classify this cat as a kitten or as an adult. So I could build a network to classify this image entirely, but maybe it's better to break your networks in small tasks. For example, here you can break your network to classify, okay, I have a global classifier that will classify this image as an animal image or a plant image, and after that I can just classify this animal image as a kitten or as an adult. So you can simplify your network, and maybe you can have better results doing that. Another thing in biology, I think, it's bias, and we are surrounded by bias everywhere, and here is no different. So when we train a network, we need to fill this network with data, and sometimes we need to classify. And when we fill this, sometimes we don't have enough data for one of the classes, but we have a ton of information for the other classes. And if we fill this network during training with unbalanced classes, we can add bias here, because this network will be more toward to segment lemons than cats, for example. What we can do here in this case, we can just remove the samples when we have too much, but there is a problem when you have to fill samples. And I know, and everyone here as a cell biologist know that data collection and creation is expensive in many forms. So what we can do using computational approach is just picking up the data that we already have and adding some noise or blur or playing a little bit with hue or crop this image. So every time that we modify an image like that, we are creating a new image set. And if you do this with your raw data and the annotated data, you will increase your data sampling. And why is data so important for training a network? Just because if you have few data, for example, here we just have 15 points, the training process will just learn too much about your data and will be super specialized to recognize just your data. And when you query your network, probably the network can fail because the new data is a little bit out of that model. So how do you resolve that? Or, sorry, and we call this overfitting. How do we resolve overfitting with just more data? So if we train the same network or the same model with just more data, so now the model will learn these little variances of your data and will fit the model. So because this is such a hot field, AI and validation and image analysis, incredible bioimage analysts just put together these two beautiful papers in metrics and recommendations for image analysis validation and understanding metrics related pitfalls in image analysis validation as well. And if you're starting this field, I really recommend you to do this and to read this kind of paper on that. With that, I like to say what I'm expecting for this field in the next few years, or right now actually. Ricardo Henrique's group just published a paper on that. So what we are expecting in this field is just what we call self-driving microscope. And what that means that the microscope or the image acquisition part of the microscope will use this deep learning approach or this machine learning or this AI approach to tell the microscope how to acquire the data. So with that, we can improve the sample health and also improve the image information that we are acquiring right now. With that, I'd like to thank you all, and especially USP and ICB, and also the core facility to support research. That's the place where I work. Irene, and I also recommend you to reach out and to be member of the Latin American bioimage community. That's a growing community to spread the bioimaging across Americas or the Latin America. And thank you.